So, coming on to canalisation and its relationship to quantitative genetics, variation at either side of the wild type. So Waddington did an experiment using two stocks from his assimilation experiments, which differed in the size of the cross-vein interruptions, and two stocks with a different addition of extra vein material to the second cross-vein and the wild-type flies. So when he crossed the two lines with different levels of vein interruption, he found that the inheritance of the size of interruption was additive. He found the same result when crossing the two lines with extra vein material. So the effects were also additive. However, when he crossed the lines on either side of the wild type, he found very different results to additive effects. The wild type therefore conceals a large range of dosages of vein-producing genes compared to any of the other genotypes. This means that when the buffering capacity is exceeded, so getting out of that valley from the epigenetic landscape, then the phenotype is altered and more or less reflects the actual dosage of genes contained within them. However, you can get some complications because the quantity of wing vein material can be arranged in different ways. You can either have a complete cross vein, same amount of vein material into interrupted cross vein with a piece of extra vein attached. So the amount of cross vein character is not homogeneous. Instead, it's more than one participating developmental pathway, with each of the constitutive pathways of the character being canalised. This is according to Waddington. So considerable interaction must occur between the genes. So in conclusion, this is Wikipedia. The canalisation metaphor suggests that phenotypes are very robust to small perturbations for which development does not exit the canal and rapidly returns down to the bottom of the canal with little effect on the final outcome of the development. However, if the perturbations have a magnitude exceeding a certain threshold, then the phenotype the ball will break out of the canal and move the developmental process into uncharted territory. Strong robustness up to a limit with little robustness beyond is a pattern that could increase evolvability in a fluctuating environment. Genetic canalisation could allow for evolutionary capacitance, where genetic diversity outside of the canal accumulates in the population over time, but it is sheltered from natural selection because it does not normally affect the phenotype. This hidden diversity could then be unleashed by extreme changes in the environment or by molecular switches, releasing previously cryptic genetic variation that can then contribute to a rapid burst of evolution. So I'm now coming on to an aspect of canalisation. So developmental buffering is the constant phenotype of organisms carrying different alleles of gene relevant to the traits under study, or that have been reared under different environmental conditions. This includes developmental stability and canalisation, but there is a debate as to whether developmental stability and canalisation are two separate mechanisms. It could affect the rate of evolutionary change. Now, one of the few identified mechanisms for buffering is the heat shock protein, HSP90. The effects of HSP90 depend on the genetic background, and if it's inhibited, much more genetic variation is observed. Through storing genetic variation not exposed to selection normally, HSP90 is perhaps a capacitor for evolution. However, mixed results in this study were obtained, so some of the results don't support the idea that HSP is a general capacitor of phenotypic variance, so no increase in the amount of variation, whilst other results, in particular the increase in shape variation in HSP90 mutants compared to the wild type, so that result supports the idea that HSP90 allows buffering of traditional constitutive traits and threshold traits, so in the case of discrete traits. But there may be too weak statistical power, so the evidence is inconclusive. Looking at the effect of experimental reduction in HSP90 activity on the variation in Drosophila melanogaster wing shape, and the relationship between canalisation and developmental stability can be investigated by comparing the patterns of fluctuating asymmetry and individual variation. If the patterns are different, then they are two separate processes. Else, highly significant patterns, there's no evidence for separate mechanisms, so studies including Klingenberg papers support either, really. Brooker et al. 2006 suggests developmental buffering mechanism, the architecture of genetic regulatory networks, which is responsible for gene expression. So that's it.